No, it's not working. Can you see it?
That's all I can do. This channel is off.
Try again. Refresh it.
Push play?
Yeah.
Can you see it?
I have an ad right now. I have an ad right now.
Oh, like it's making noise.
Yeah, it's working.
Okay.
Can you send it to his uncle and grandpa? Should I email it to you or text it to you? I G-chatted to you.
All in a day, stop the phone, and just are you gonna take it?
What?
On Facebook?
Yeah. Am I taking somebody's spot? I'm not tagging him. I just updated that Facebook post. If you want to put it on like the editor Facebook. If you want to put it on Facebook, like, do you want me to put it on his profile? I just put it on that link.
Yeah, put it on his page. If you don't mind. Kill my phone. So I just said, is that the same link you texted me?
Yes. I don't know how to say it.
Is it working now?
I think so. It's on a delay, but it's working.
Can you send me that link? Can you text it to me or send it to my phone somehow?
Sure.
Getting the new link ready. This student's not a lot of faculty.
You always come late.
I like it better this way. We're waiting for Dr. Valley.
Should I sit up there? I'm like, yes.
Can you also paste it directly to my Facebook as well?
I'll leave the seats in the back. Are you going there? Then I'll go behind.
That's why I didn't do the first one.
Are you gonna sit here?
Can I get you to bring me my charger from the back, please? And the headphones.
Is it working?
I think so. It's on a little bit of a lag.
That's fine. It doesn't matter. They will know.
Thank you. No. unblock people's views?
Yes, that's totally it.
I know as well. I know they did these resort regulars. There were these guys and they were all awesome. Like just dancing and practicing the dances. So I was standing in front of two girls. I was like, oh my gosh, it's right for you. And they were like, oh, you can sing. That's fine.
Alright, I'm not 5'11".
At this point, I wouldn't say anything, so don't worry about it. I should be on that was point of yeah, it's a cool, there's such a nice connection. It's actually a question I was really pressing question. Oh, yeah, I'm going to say that because I've tortured you.
That on as well.
Okay, guys, it's my pleasure to get to say a few words to introduce Joel, but I don't want to take up too much time.
Did you guys coordinate your outfits?
We did. we always do. So Joel graduated from the University of California, Santa Barbara in 2010, and then quickly, soon after that, came to the Human Genetics program at Hopkins. He originally joined Ronnie Cohen's lab, but in Joel's second year, Ronnie was recruited to Toronto, and thankfully for me, Joel decided to stay in Baltimore, and I was able to recruit him to my lab, which actually wasn't much of a lab at that point because we hadn't even gotten space. But Joel has been a great partner in helping set up the lab, and it's been a very fun collaboration working with him. So I just wanted to emphasize one thing. During these 3 years that Joel has worked in my lab, he has been able to show that Kabuki syndrome is a potentially treatable cause of intellectual disability. And his work shows, and you'll see the work, that there are at least 2, maybe 3 different ways that one might be able to treat that condition postnatally. So I think that's very exciting. He has 1 published paper, and he has a manuscript that's ready to submit. So I think these are great achievements, and he has done this by being a very smart guy, a very, very competitive guy, I can tell you that. Very hardworking, but first and foremost, very focused. He's always been very, very focused. So it's been a pleasure. It would be a loss to see him go, but we're very happy to say that he has accepted a position at Stanford in the laboratory of Thomas Rando, which I think is going to be a wonderful environment for him. So I want everyone that can should join us. We will be hosting a party for him on the 2nd floor of the MRB after this talk. But without further ado, I just want to present Joel Benjamin, and he's going to show you some of the great work he's done in the last 3 years. Thank you.
Lights. Are you ready? All right. So thank you, Hans, for that nice introduction. I'll try to live up to everything you promised. Hi everyone. My name is Joel Benjamin, and I'm going to tell you a little bit about my thesis work I've done in Hans Bjornsson's lab entitled "Kabuki Syndrome: Reversing Intellectual Disability by Promoting Open Chromatin." Now I know I've lured you all in here with the promise of a translational talk, but I'm actually going to go to the basics first and just get into some of the basic biology and just give a brief background on epigenetics. So to start, I'll be briefly reviewing chromatin and the compaction of the genome. So each cell has to fit about 3 meters of DNA into a relatively small nucleus. This is done by wrapping DNA around histone proteins to form a structure called a nucleosome, and then this is folded on itself. And this folding of DNA and histones is called chromatin. This packaging plays a large role in gene expression, and this regulation because the region-specific compaction of the genome allows for different genes to be accessible. If the DNA is accessible, it's said to be in a state of open chromatin. If it's not accessible, it's considered to be in closed chromatin. One way in which the expressions of genes is regulated is by chemically modifying DNA with the DNA's association with these histone proteins to either promote open chromatin or to close it. The study of these modifications that are not in the DNA sequence but affect its expression is called epigenetics. What are particularly interesting about these modifications is that they are malleable either by endogenous or exogenous perturbations, and these marks can either be added or removed reversibly. And there's two main areas of field in the study of epigenetics. One is the modification of DNA itself, such as the methylation of cytosine in DNA, and the other is the modification of the histone tails. And for this talk, I'm going to be mentioning different kind of methylation, but everything is going to be about histone tail modifications. Nothing is about DNA methylation. All right, so we can look more closely at this nucleosome structure. And it is made up of 8 core histone protein components that are bound by DNA. And each of these histone proteins has a tailed region that has amino acid residues that stick out. And these residues can be chemically modified by different chemical groups. And these different chemical groups can have different functions that can open or close chromatin, as well as recruit other proteins to carry out these jobs. All right, and these marks aren't added or removed just by chance, but they are done by specific proteins. And there are 3 main classes of these proteins: writers, readers, and erasers. So writers add marks, erasers remove them, and readers interact with place marks and other machinery to carry out downstream gene expression consequences. Now, what's interesting about these modifications is that many of them have well understood function as to whether their presence opens chromatin and activates gene expression or closes it and represses it. So here are just some common marks in open chromatin and common marks seen in closed chromatin. So in open chromatin, we have the histone 3 lysine 4 trimethylation, as well as acetylation and multiple residues on histone 3 and 4. And some common seen closed marks are histone 3 lysine 27 trimethylation and histone 3 lysine 9 trimethylation. And these are some of the most well understood ones, but then just looking at a diagram here seeing other histones, you can see there's many other marks that have been studied that are known to have closed or open chromatin. And this can all get a little bit confusing, so for this talk, I really am just going to focus on this one mark here, H3K4 trimethylation, which is histone 3 lysine 4 trimethylation, which means the 4th lysine residue of the histone 3 tail gets 3 methyl groups. And that's very strongly associated with open chromatin. So the reason that I'm focusing on that mark is because of its effect in Kabuki syndrome, our disease of interest in the lab. So Kabuki syndrome is a rare autosomal dominant Mendelian disorder, and the symptoms include characteristic facial features which were thought to resemble the makeup of Japanese Kabuki theater. At least that's how it's got its name, and these include inversion of the lower eyelid, downturned ears, flattened face, and arched eyebrows. I guess they thought it looked similar. You can make up your own decision. And also are seen are growth retardation, early hypotonia, immune deficiency, currently of unknown etiology, as well as several other rare phenotypes seen in a subset of the cases. However, what makes it, what's most important to us is the fact that this is also an intellectual disability, is the main phenotype seen here. And intellectual disability affects up to 3% of the world's population, and it's such a devastating clinical diagnosis because there currently isn't much of a treatment. So the reason we are particularly interested in Kabuki syndrome then is because of the gene that causes it, KMT2D, which is involving the epigenetic machinery of the cell. And because of the malleable nature of epigenetics, may allow for some malleability here and potentially reverse the intellectual disability, or at least that's the hypothesis that we were working on. So now that I've mentioned what we think the gene involved in Kabuki syndrome is of unique interest, let me tell you a little bit about it. So KMT2D stands for lysine methyltransferase 2D, and it's a writer, as I said, for this open mark of H3K4 trimethylation. It's the main mutation in Kabuki syndrome, and it causes dominant loss of function mutations, also known as haploinsufficiency. And just to give you an example of how this would work, you could have in closed chromatin, you could have a histone 3, the lysine 4 does not have trimethylation. And in open chromatin, we have trimethylation placed, and this is mediated by KMT2D. In Kabuki syndrome, where we have a half loss of KMT2D, we would expect to have much less open chromatin as we're not adding enough of this mark. At least that's our hypothesis. So therefore, we hypothesize that normally there is a balance in chromatin between open and closed states for the important target genes of KMT2D. And we hypothesize that Kabuki syndrome is marked by a dynamic imbalance in which because we can't add this open mark, we're shifted to more closed chromatin states. And we think that this imbalance underlies the pathology of Kabuki syndrome. So to test this hypothesis in vivo in a system that could best translate many of the human aspects of the phenotype, we characterized a mouse model of Kabuki syndrome, which we'll call KMT2D BGO, or just KMT2D mice. So now I'm going to come to my outline. The way this is going to work is I'm going to put a point up, and then we're going to fill the outline as we go along, and then at the end we'll have a summary to look back on. So the first question is, does this mouse model KMT2D-BGO effectively model Kabuki syndrome, and then does it also give us some insights into the pathology that were not currently understood. So first, to get to the nitty-gritty, KMT2D-BGO is a heterozygous knockout of the functional methyltransferase domain of the KMT2D protein. So this domain is made up of exons 51 through 53. What we've done here is it's just a simple knockout where we've removed these exons and replaced it with a BGO cassette, which is inert. And then we confirm this knockout by qPCR. As you can see, primers for an upstream exon, exon 20, are equal levels in both the wild types and the KMT2D mice. And exon 52, which is part of this deletion here, we only see half expression in the mutants in the KMT2D mice compared to wild types. We also can see here from conventional PCR that KMT2D mice amplify both primers for the controlled DNA band, as well as BGO, and wild types just amplify the control. And then one more control, we looked at whether this protein was made. So we did an immunoblot against the BGO cassette, and we saw in KMT2D mice that this protein is being expressed. We see the presence of this BGO cassette. It's not present in wild types. All right, so the first thing that we did when we began to phenotype these mice was to see if they resembled Kabuki syndrome in any meaningful ways. So one aspect that we noticed was that these mice seem to have similar facial profiles to Kabuki syndrome. So you can see here in this patient, I mentioned the flattened face, a shortened flattened face. And you can see here in the mouse, the skins have been removed. This is wild type, this is mutant. You can see the shorter, flatter face. And then here are CAT scans. You can see in a wild type, or in a healthy patient, you can see a normal facial profile. And in a Kabuki syndrome patient, it appears, the best way to describe it would be sort of an underbite. And when we look for this feature, we began to see it in the mice. So here's a radiographs from wild types and KMT2D mice. It's a little bit hard to see here without looking closely, but looking at this overbite here, we thought that we saw a phenotype. But to confirm this phenotype is real, we had blinded participants just separate groups of mouse radiographs just based on this overbite phenotype. And when they did that, just separating, they were able to very easily separate between wild types and KMT2D mice, suggesting that this phenotype is real. Additionally, we saw many other phenotypic features that resembled Kabuki syndrome in several ways. These are not going to be the focus of the talk, but I think it's interesting to look at them. And I think not only does it confirm that KMT2D mice resemble Kabuki syndrome in some way, but they also suggest that this may be a good model for studying other aspects of the disease down the road. So these include growth retardation. You can see at every time point measure that the KMT2D mice in yellow here are smaller than the wild types. And at old age, at 12 months, we actually see enlarging of this weight gap. And this we believe is due to obesity resistance. You can see a wild type mouse and you can see a KMT2D mouse and you can see how much thinner it is. We also see hydrocephalus, which is not a normal, or it's not a common phenotype you see in Kabuki syndrome, but there are case reports of it. And while it's very rare in mice, we see it at a much higher proportion in our KMT2D mice. And what this is basically is a buildup of cerebral spinal fluid in the brain, which enlarges the ventricles and expands the skull. We also, again, see the craniofacial abnormalities, which you can see here that from the front view, you can see the KMT2D mice have a flatter, kind of rounder face. We also looked at immune deficiency, which is a large problem in Kabuki syndrome patients. And here we're just looking at B cell markers. This is B220. We see about a half loss of that from FACS comparing KMT2D mice to wild type. And then finally, we also noticed lymphoproliferation, which results in an enlarged spleen in the KMT2D mice related to wild type. And we believe this is related to the fact that the spleen plays a larger role in the immune system of mice. So now getting back to our main story here, which is the study of the potential epigenetic defect, we hypothesized that there would be a global loss of H3K4 tremethylation. So what I have here is data from ChIP-seq that was performed by Li Zhang in the lab and analyzed by Dr. Casper Hansen. And just to explain to you how this works, we have ChIP-seq against H3K4 tremethylation. The x-axis is the genomic location, the y-axis is the fold expression. And every red dot you see is a ChIP-seq peak. And it's a comparison between the KMT2D mice and the wild type mice. Any peak that's above 0 is higher in the KMT2D mice. Any peak that's below is lower. And I think you can appreciate from this graph, and this is just a global look, and we still have to investigate the genes that are underneath these peaks that would be expected to be less expression, that you can see a global shift towards less expression of H3K4 tremethylation peaks. And I should mention that all of this data is taken from blood. And we're just using this as a global look to see, not at any particular tissue, but just to see if there's global decrease of these peaks. So we next asked, where is KMT2D expressed in the brain the highest? Because we're still trying to look for a cell population that could be relevant to the intellectual disability phenotype to hope to characterize in the mice. So I did immunofluorescence staining across the entire brain to see where KM2TD showed high levels of expression. And we saw that one area of particularly high expression was the dentate gyrus of the hippocampus, which is here and here. And so one reason this was particularly interesting to us was because this is one of the few areas of adult neurogenesis where neurons are born in the brain. And it's thought to be important for memory and learning. So considering KM2TD's role as a writer for H3K4 trimethylation, we asked what the levels of H3K4 trimethylation were in this dentate gyrus, specifically the granule cell layer, which is these arms here. So when we looked at that, I first did immunohistochemistry just to see if I could see a difference, and top is wild type, bottom is KMT2D. And I think you can appreciate that the wild types are darker, and that the darker it is, the more H3K4 trimethylation antibody is present. However, it was a little bit hard to quantify from the immunohistochemistry, so we did immunofluorescence. So just to explain these images, blue is DAPI, and DAPI is a stain that will go into every cell and bind to the DNA and color that blue. So every single cell will stain for DAPI, and we use this as a control to see which cells, just to see which cells are present. And then we have an antibody against H3K4 trimethylation, which here is pinkish red. And I think you can appreciate here in the wild type compared to the KM2TD mice how much more bright and robust the expression is of H3K4 trimethylation in the structure compared to the mutants. And then when we quantified this looking at the fluorescence intensity, we saw a significant decrease in the KMT2D mice compared to the wild types. Because of the dentate gyrus of the hippocampus is important for forming new neurons, we wanted to see if neurogenesis here was affected. So we used the expression of a protein called double cortin, which is expressed during intermediate neurogenesis. And the reason that we picked this protein was because any neuron that's expressing double cortin is one that's going to be expected to be undergoing neurogenesis. So what we saw here on the left is a wild type, here is a KMT2D mouse, and you can see looking at the greenish yellow color, that's double cortin. You can see there's pretty robust expression, and this is in 1 to 2 month old mice of double cortin here, right in the border of the granule cell layer, and we see a pretty big loss of that in the KMT2D mice. Just a more zoomed in picture, you can appreciate how much thicker and robust the cell layer is in wild types and how much thinner it is in KMT2D mice. And when we quantified the area of the cell population, we saw a significant decrease here. So because of the defects seen in the dentate gyrus, a structure in the hippocampus, we wondered if there would be any hippocampal memory defects. So we did a lot of different behavior tests, but the one that we really settled on was the Morris water maze, which is considered the gold standard assay for hippocampal memory. So for those unfamiliar with the test, I'll just go over it quickly. So the goal of the test is to train a mouse to find a submerged platform hidden beneath opaque water, usually painted with white paint. And then it uses visual cues around the room to help remember where the platform is, and over the course of the training find it. So the way that we did this was the first 3 days was something called flag training, where a visible flag is placed on top of this platform. And then the mice are trained just to get to the visible platform, just to make sure that they can get there. After that training is complete, we do 5 days of hidden platform training where the flag is removed, the mice can't see the platform, and they're put in the tank at various locations for multiple trials per day for 5 days, and trained to find this platform. And at first they obviously can't find it because they can't see it, and then as the trial goes on, they're able to get there faster and faster. And then the final day, to see how well they've learned where this platform is, we actually do something called the probe trial, where we completely remove this platform, and we give each mouse 90 seconds to swim, and we see what their association is with the platform. And there's lots of different measures that can be used here. Some people look at the time in the quadrant of the platform, some people use distance from the platform. The measure that we used was the number of times the mice crossed the platform, and the reason we picked this was because there was a paper that said that this correlated very nicely with neurogenesis levels. So just to show you, this is representative data. I point out the platform, it's a little bit hard to see here. You can see in wild type mice, these mice swim in a regular fashion. They tend to cross over either very close by or over the platform zone. And the KMT2D mice seem to swim more aimlessly and cross over it less. So when we quantified the number of crossings from several cohorts of mice, we saw a significant decrease in KMT2D's crossing of the platform compared to wild types. So coming back to our outline, coming back to the question, does KMT2D geo-effectively model Kabuki syndrome and give pathological insights? I think the answer is yes. So we see that it shares several phenotypic and physical features with Kabuki syndrome. We see decreased open chromatin, decreased neurogenesis, and also from the ChIP-seq we saw decreased peaks, and also hippocampal memory defects, which in some ways are similar to patients. However, as I stated earlier, the goal that we set out to start with was not to just characterize the mouse, but also to see if we could reverse these deficits. So let me show you the hypothesis behind this goal. So, simply put, we previously thought and appeared to confirm that KMT2D mice, and as a proxy for Kabuki syndrome, have an imbalance of open chromatin due to a decrease of H3K4 trimethylation. We therefore hypothesize, because chromatin states are modifiable, and that it appears that the imbalance is related to the phenotypes, that if we can somehow promote open chromatin, we may be able to restore the balance of open and closed chromatin, and then maybe reverse the downstream phenotypes postnatally. So the drug that we chose to use is called AR42, and I've shown it here. And it's part of a class of drugs called HDAC inhibitors, and you may have heard of these before. And HDAC inhibitors have recently been used as treatments in many diseases, including cancer, epilepsy, and even the intellectual abilities in certain diseases. And while specifically the name is derived from inhibiting the erasures of acetylation, which is known as an open mark, because open chromatin systems seem to be tightly linked together, they frequently also increase H3K4 trimethylation, a mark of open chromatin. And also we had known previously that AR42 does increase our mark of interest, and that's one of the reasons why we picked it. So I'm just going to show you the treatment strategy we used in vivo for the mice. So we did a 2-week treatment at several doses, and this was given by oral gavage done by Yi Chen from the Thesis lab. And this was guided by previous in vivo studies using AR42 from a previous group. And then we performed behavior tests right at the end of this trial. And then we sacrificed and perfused the animals on the 15th day after the start of treatment, and then section and stain the brains to do analysis. So the first thing that we wanted to look at was double coordinate expression to see what effect AR42 would have on neurogenesis. So just as I've shown before, here's a wild type and here's a KMT2D mouse, and you can see here the greenish yellow is double coordinate expression for showing us neurogenesis, and this is just the vehicle, so this is the same as no treatment. And so first I wanted to look at just the highest dose. If we just blasted it with this drug, what do we see? And what we see was not super promising. We saw a pretty big decrease in both wild types and KMT2D mice for double coordinate, so that was troubling. But then we looked at the lower doses, and what we saw was actually quite interesting. So the wild type levels don't seem to change much at these doses, a little bit decrease, but don't change much. But the KMT2D mice at about 10 mg per kilogram per day of AR42 seem to have a pretty nice increase of double coordinate expression at this dose. And when we quantified this, in the same way we had quantified it earlier, we can see at this dose we have a significant increase compared to vehicle at 10 mg per kilogram per day of AR42. And there's no longer a significant difference between the wild type and the KMT2D mouse at that point. Now that we had found a dose that rescued neurogenesis, we wanted to make sure that we had actually opened chromatin and increased the H3K4 trimethylation, as our hypothesis was. So again, here is just vehicle. We can see the same decrease in this mark in the KMT2D mice compared to wild types. And then on this dose of AR42, we actually have a massive increase in the KMT2D mice and also an increase in the wild types. And when we quantify this, we see this. So we see a significant increase here, which takes it up to wild type levels. Although it's important to point out that the wild types do better as well, and I'm going to remember that because I'm going to come back to that later in the talk as to why that might be. So furthermore, coming back to the chip-seq data, this is the graph I showed you before. We wanted to see what effect this dose of AR42 would have on the chip-seq. So here, again, is what I showed you before, just the regular untreated KMT2D mice compared to wild type. We saw a decrease in peaks, and then when we compared the drug treated to the wild type, you can actually see this correction back, and actually even a slight overcorrection. I think you can appreciate how many more peaks are above zero here, showing an increase back to wild type levels. And then finally, and most importantly, we wanted to see what effect this dose of AR42 would have on the BGO mice's performance on the water maze. So we saw that these mice treated with AR42 show a significant increase in platform crossings compared to their untreated KMT2D mice, and it brings them back to no difference compared to wild types, although again, pointing out that the wild types increased as well. And I'll come back to that as well. But what we've demonstrated here is that asking this question, can we do a pharmacological treatment to rescue KMT2D mice? What we've demonstrated here is that based on our hypothesis of excessive close chromatin underlying the phenotypes of KMT2D mice, that using a drug that promotes open chromatin rescues these phenotypes to some degree. And while correct human dose levels in the treatment window would need to be established if we ever wanted to fully translate this to Kabuki syndrome, these data provide strong evidence that, as a proof of principle, that this treatment strategy could be used in Kabuki syndrome at some point down the line, or at least that this type of strategy has potential. However, there is one, if we want to talk about the clinic, there is one problem with this. So AR-42 is normally used as a cancer therapeutic, and it's actually a rather strong drug. And it's going to be pretty difficult to transfer that sort of drug to the clinic for a non-lethal disease. So we were trying to think of other ways that we could have a similar rescue, but avoid such a powerful drug. And so the idea that we settled on was, and this was kind of wishful thinking, but this was the hypothesis that we worked on, was instead of using a drug, could we use a dietary treatment to promote open chromatin in a similar way and show a similar rescue? So you're probably wondering what kind of dietary treatment would be able to recapitulate the effects of the strong HDAC inhibitor. So to answer this question, or at least the starting point for us, was from a paper. We got this from a paper that came out from Eric Verdin's group in 2013, in which they demonstrated that the ketone body beta-hydroxybutyrate, which is naturally produced through the body, is an HDAC inhibitor, histone deacetylase inhibitor. So the reason this was exciting for us was because of the way that beta-hydroxybutyrate is naturally produced in the body. So for this, I apologize, I'm going to take a slight detour into metabolism. So during the normal fed state, when glucose is high, it is broken down into various stages into acetyl-CoA, which is then used to power to run the TCA cycle to produce ATP. And then during times of low glucose, also known as ketosis, fats are preferentially broken down to create acetyl-CoA for the TCA cycle. And then this step that breaks down fatty acids to acetyl-CoA is known as beta oxidation. And during the step of beta oxidation, this is where you produce the ketone bodies, acetone, acetoacetate, and beta-hydroxybutyrate. So such a diet that promotes the preferential breakdown of fatty acids by limiting glucose and carbohydrate levels and results in the production of ketones is known as a ketogenic diet. And just kind of to summarize this, to drive this point home, in short, what appears to be here that we have a dietary regimen that can naturally produce an HDAC inhibitor, which is the same type of drug that I had previously shown has a decent effect on our mouse model of Kabuki syndrome. So that leads us to my next hypothesis here, which is based on our previous hypothesis, we therefore predicted that treatment with the ketogenic diet, which would increase the levels of endogenous beta-hydroxybutyrate, the HDAC inhibitor, would be able to promote open chromatin in a similar manner in our mouse model of Kabuki syndrome, and potentially so maybe a similar rescue to AR-42. So first in vivo, I'm not showing the data, but I was able to show that beta-hydroxybutyrate does in fact increase our open chromatin mark in cell culture. I'm not showing that data here, but we were able to see that. And then here's just the treatment strategy for the in vivo work with the mice. So we did a 2-week treatment with the ketogenic diet. So it's a paste that we bought from a company. It smells exactly like Cold Stones, but it tastes awful, says Gio from my lab. I don't know why he had to have a whole spoon, but that's up to him. So on this treatment, we in the same way we did with AR-42, we performed behavior tests on the diet, treated mice towards the end, and then we perfused, sectioned, and stained, and analyzed the histone-neurogenesis effects. So now I'm just going to walk you through the same things that we looked at with AR-42 and see what rescue, if anything, the ketogenic diet had. So the first thing that we want to do, before I get to that, is we had to make sure that beta-hydroxybutyrate actually got increased. If we're claiming that this diet is going to increase it, we should probably check that it's increasing in vivo. So in this graph, we have just regular diet-treated animals and ketogenic diet-treated animals, and we can see a significant increase in beta-hydroxybutyrate as measured from the urine. So the ketogenic diet is doing its job. So now let's look at the different phenotypes that we saw that were rescued by AR-42 and see what happens with the ketogenic diet. So first we looked at the levels of H3K4 trimethylation in the dentate gyrus granule cell layer. And what I want to point out here is we do see a significant rescue for the regular diet-treated KMT2D mice compared to the diet-treated mice. We do see a rescue, but it's a little bit less than what we saw with AR-42, and there's no effect on the wild types. And if you remember before, AR-42 was able to increase both of these levels much higher, but it appears that this is only affecting our specific KMT2D mouse model, not regular wild-type mice. So we next looked at double coordinate expression to see if we were able to rescue the neurogenesis as well. So again, you've seen this, just the regular diet-treated green is double coordinate, and then interestingly, we were able to see a very nice rescue with just 2 weeks on the ketogenic diet for the KMT2D mice that we're able to, actually the results look very similar. We see a significant increase here that were very similar to what we saw with that optimal dose of AR-42. However, we wanted to really be sure that this was the case, so we decided to use an alternative method as well to measure neurogenesis. And let me explain how that works. I haven't mentioned that yet. It's called EDU staining. So EDU is a molecule that interpolates into DNA during cell divisions, and it labels newly divided cells. So what you can do is you can inject EDU into a mouse for as many days as you want. In our protocol, we did 7 days in a row. And then if you sacrifice the mice and stain the tissues against an antibody against EDU, you can see which cells have newly divided. If you look at the dentate gyrus, any cell that's newly divided by definition is one that's undergone neurogenesis. So this is, instead of just looking at a marker of a cell at a snapshot, we're actually looking at across a 7-day time point, how many cells have newly divided. So it's a little bit different. So we first wanted to see if we could recapitulate what we saw with double court, and we could see that. So here, EDU is these green dots here. It's a little bit hard to see in the back, I apologize. And you can see a much higher level of EDU positive cells in wild-type mice compared to KMT2D mice. And then interestingly, we actually saw very similar rescue to when we looked at double court, where we see an increase in the KMT2D mice expression of EDU positive cells on the ketogenic diet. And when we quantified this, and this is much easier to quantify, just counting cells, we actually see a almost full recovery for the KMT2D mice, and there's no significant difference compared to wild types. And then finally, we wanted to look at the most important measure for us, which is the behavior test. We wanted to see whether the diet treatment had a beneficial effect similar in the way that AR42 had. And then again, looking at the platform crossings during the probe trial of the Morris water maze, we see a full rescue for the KMT2D mice. It gets them back to wild type levels. However, and I've pointed this out several times, there is no effect for the wild type. And I'll come back to that, but again, it appears that the ketogenic diet only showed a good increase for the KMT2D mice. So there was one more loose end we had to fill out for this story. So if we're claiming that the ketogenic diet is acting through beta-hydroxybutyrate that's being produced, we should show that beta-hydroxybutyrate actually is what is causing this effect. Other people have used the ketogenic diet, and it does cause massive metabolic shifts. And people have had many different claims as to what the ketogenic diet does or what aspect of it causes rescues. So we want to say, okay, if we just go to a simple experiment, if we just give plain beta-hydroxybutyrate to mice, can we show a similar rescue? I'm just going to show you data from neurogenesis. So the first thing I did was I had to establish what dose to use. From a dose response curve, I'm not going to show you the data for. I found about 10 millimolar per kilogram per day, one injection. Seems to mimic about the high point of what a ketogenic diet gives you for beta-hydroxybutyrate. So I gave this injection for 2 weeks, just as the ketogenic diet, and I saw what effect this had on neurogenesis. So what we saw here was kind of interesting. So we did see a significant increase for the KMT2D mice, but it doesn't get us back to wild type, and it just, you don't get as much of a recovery as we did with the ketogenic diet. So something about just giving this injection of beta-hydroxybutyrate was not enough to rescue it. So I was thinking about this, and I figured this could mean one of two things probably. So either there's some aspect of the diet that's also having a rescue that we didn't consider. The other alternative is the one injection I'm giving wasn't enough to have a rescue. You know, I see a spike, but that's not lasting enough through the whole day, while the ketogenic diet is giving us constant supply. So to test the second diet, I just did an experiment where I gave, over the course of a day, I gave one injection in the morning to a group of mice of beta-hydroxybutyrate. I took mice on the ketogenic diet and then mice just on a saline control, and I took urine at different time points, and I measured beta-hydroxybutyrate throughout the day. What I saw was actually pretty interesting. So just to orient you to this graph, the x-axis is time. Blue is mice that were injected with this one injection of beta-hydroxybutyrate at 9 in the morning. The red is just a saline control, and green is the ketogenic diet. So you can see the ketogenic diet mice, they fluctuate throughout the day, but they're always kept at a relatively high level of beta-hydroxybutyrate. What's considered ketotic is above 1 to 2, so these are always ketonic. And you'll see with the beta-hydroxybutyrate injection that you do see this spike. We are able to increase this up to the ketogenic diet levels, but this actually quickly subsists and then doesn't spend as much time throughout the day in a ketotic level. So I just did a quick estimate of this, and I estimated that this injection, they only spend about 10% of the time with the beta-hydroxybutyrate level that matches the ketogenic diet, and that's including the fact that there's other 12 hours that aren't accounted for here. So what we wanted to do then was we asked, is there any way we can supply a more constant level of beta-hydroxybutyrate to these mice? So what we settled on, or at least attempted to try first, was using osmotic pumps. So the way these things work, there's these little pills that have a reservoir in there, which you can put in an extremely concentrated amount of drug. You implant this into the back of a mouse, and it slowly diffuses the drug over the course of 2 weeks, and it gives an even flow rate. And so I was able to concentrate, the maximum concentrate that I was able to make was 3 grams per ml of beta-hydroxybutyrate before it was insoluble, which came out to about almost the exact same dose as the injection we gave, but instead of at one point, it's spread out equally through the day. And just to show you what this looks like, here's the pump installed in the back of the mouse. And we did this for 2 weeks, And what we saw here was that we actually did see a significant increase of beta-hydroxybutyrate in the beta-hydroxybutyrate given pumps compared to saline controls. The problem is, if you look at the y-axis, we're not getting up to ketosis levels. So we're giving a constant supply, and it is higher, but it's not going to be high enough. But since we are giving a constant level, my thought was if I can supplement this at several different points throughout the day with injections of beta-hydroxybutyrate, maybe I can better mimic the ketogenic diet beta-hydroxybutyrate level. So that's what I did. I gave injections at 3 time points throughout the day. And then again, the same colors, green is ketogenic diet, blue is beta-hydroxybutyrate, red is the control. And you can see here, when I give these 3 injections, while there are spikes and peaks, they do spend much more amount of their time above the 1 to 2 millimolars of beta-hydroxybutyrate throughout the day. And then when I just did a quick estimate, I estimated about 40% of the day they spend in the ketosis state, or a beta-hydroxybutyrate level that's similar to that. So I felt that I was giving a much more amount of the drug constantly throughout the day. So when I did EDU staining of these mice, what I found was that I was able to actually recapitulate almost exactly what I had seen with just the ketogenic diet, where I see a full rescue back to wild type, and this graph looks very similar to what I saw for EDU with the ketogenic diet. So when I give lots of beta-hydroxybutyrate, I'm able to have a rescue that's very similar to the ketogenic diet. So as we've seen with the ketogenic diet, while it may not have shown as strong a response as AR42, it seems to show similar rescue for the phenotype seen in KMT2D mice. And this seems to be because of the presence of increased levels of beta-hydroxybutyrate, as demonstrated from our sufficiency test at the end. And as you can imagine, the implications of this study are pretty big. That not only can we modify, just through changing the environmental interactions, we can modify the epigenome, but the impact this could have on the clinic, getting away from a drug and just giving a dietary treatment that's already been approved, and even at Hopkins they have a whole center set up just for the ketogenic diet, is really appealing to us as a lab that eventually wants to try to get into the clinic. So not only were we able to show that a drug was able to potentially reverse some of the aspects of the intellectual disability, but we were able to show that through a similar mechanism, with a similar idea, we were able to have a similar rescue just from the diet. So this was great. This was really exciting for me. This data here is part of my next paper. But there was one more thing, and I kept bringing up, there was one more thing about this that was still bothering me that I wasn't satisfied with, which actually led me to some new insights about this mouse model, which actually may give even more support to why we should use the ketogenic diet as a treatment on these mice and then potentially on patients. And if you remember from the slides, I kept pointing out that AR42 increased both genotypes, but the ketogenic diet only had an effect on just the KMT2D mice. So the question that I asked is why? Why does the ketogenic diet appear to show a genotype-dependent efficacy? So one possible answer for this, and I kind of brought this up before, is that maybe AR42 is just much more powerful. And that's possible and actually could still work with my other hypothesis, and that could totally be the case. But I'd like to show you some data that suggests an alternative hypothesis, and that alternative hypothesis may make the ketogenic diet treatment even more effective for Kabuki syndrome. So coming back to this graph, I showed you this already, and if you were paying attention, you probably had this question, which was, okay, so he showed us that the ketogenic diet increases the beta-hydroxybutyrate levels in urine more than the regular diet. But why didn't he separate this by genotype? So when I do that, we see something pretty interesting. So for the regular dry diet-treated mice, there's no difference between the KMT2D mice and wild type. But on the ketogenic diet, the wild type increases as would be expected for beta-hydroxybutyrate. But the ketogenic diet levels the ketogenic diet beta-hydroxybutyrate for the KMT2D mice go up even higher. We have a hyper elevation of beta-hydroxybutyrate. So it seems like these mice put on the exact same diet are overproducing or producing more of the active particle that we think may provide the rescue. So the first thing that we had to test here was that, do we actually have an increase in beta-hydroxybutyrate, or is this just hiding some other piece of data? So let me tell you what I mean. So the two ketone bodies, acetoacetate and beta-hydroxybutyrate, can actually chemically interconvert with each other. So it was important to see if we had an increase in both metabolites, which would kind of nullify this. It would just mean that we have an increased flux of the whole system, or if we actually have a shift here where this interconversion is shift towards beta-hydroxybutyrate. So we did, with help from Hilary Vernon from the Vernon lab, we did mass spec, and looking at the ratio between beta-hydroxybutyrate to acetoacetate, we actually saw a significant increase in that ratio. So we actually are it's not just an increase of both. We actually have a shift in this where we have an increase of beta hydroxybutyrate. And why this is interesting is because this interconversion is governed by the NADH to NAD plus ratio of the cell. So as this diagram states, high levels of NADH, if you have high levels of NADH, would be expected to drive this reaction to the right side. So which suggests here that because this reaction is driven to the right side, we could hypothesize that we have a higher NADH to NAD plus ratio. So to test this, the best thing to do would just be look at another ratio that's governed by this the same metabolite. So the one that we thought of was pyruvate to lactate, and that also would be driven to the right side here, an increased lactate to pyruvate ratio if we had higher NADH. And when we look at these levels, we also see a significant increase where we have a shift to the right where we have an increased lactate to pyruvate ratio. So I tried to synthesize all this data in my head to kind of come up with a hypothesis as to what's going on here and why I think this is important. So just coming back to what I showed before, when I showed the fatty acid breakdown during ketosis, fatty acids break down to acetyl-CoA, and then they're used to power the TCA cycle. And this step here, fatty acids to acetyl-CoA, is beta oxidation. And during beta oxidation, we have the production of the ketone bodies. I'm not showing acetone, but those produced, two of those are acetoacetate and beta hydroxybutyrate. Those are the two ones I just showed you that can interconvert. And also what I haven't mentioned is that NADH is also produced during beta oxidation. So this is all just the metabolism behind it. So now I want to introduce my mouse. This is a great drawing of my mouse. So just some facts that we know about it. So they're leaner and at old ages are obesity resistant. So here's a little bit of hand waving, but just my hypothesis about what's going on here. I believe that these mice, the reason for this is they have increased fat breakdown, and there are ways that we could test this later to test this hypothesis, but I hypothesize that they have increased fat breakdown and they have increased rates of beta oxidation. And if you give them more fats in the diet, instead of storing them, they're going to break them down. So then if we get into a system where we have increased fatty acids, like a ketogenic diet, what I would expect to happen here would be increased beta oxidation, which of course would then have more NADH. We already know what happens when we have more NADH, which is it drives the reaction to the right, which means we get higher beta hydroxybutyrate levels. So what this suggests to me is that these mice, and then maybe as a proxy as well, as a mouse, definitely, as a proxy for Kabuki syndrome, actually may be predisposed just by their own genetics or their own mutation to creating higher levels of beta hydroxybutyrate. And why this is interesting for the treatment here is that a standard ketogenic diet may be more effective for them than just anybody else because it's almost like they're tailor-made to respond to it better. So now we fill out our entire outline, so now I'm just going to come back and go over it. So I've been building a summary, and just to go over quickly, I've demonstrated that these mice model Kabuki syndrome as well as give insights into the pathology of the disease. I've shown that based on our hypothesis of an imbalance of chromatin, that KMT2D mice can be rescued in these phenotypes, including behavioral, by promoting open chromatin, and pharmacologically with an HDAC inhibitor. I've also shown that a ketogenic diet treatment can be used in a similar manner for KMT2D mice, and in fact this treatment may be especially effective for these mice and potentially Kabuki syndrome patients because of their predisposition to increase beta hydroxybutyrate levels. And then while the exact timing window and treatment length must be refined, these data present a strong evidence that KMT2D mice are a powerful model for the study and treatment of Kabuki syndrome, and that this strategy for potentially reversing intellectual disability postnatally may one day work in the clinic. Thank you. And I just want to go over my quick acknowledgments real fast. So I just want to thank everybody in the. Do I do this first, or do I ask questions first?
No, you do this.
Okay. I've never done this before. So I just want to thank everyone in the Bjornson lab, Hans Bjornson, Li Zhang, and I'll get to more of these again in my fuller.
Is that a threat or not?
It's a promise. Jill Farner, Jackie Weissman, Giovanni Caruso, and Janae Polarowski. I said that right? The graduate students in my lab and my old undergrad student Hanso Cho. From the Thesis lab, we had lots of help from Dr. Hal Beetz, especially on our first publication, Dr. Elizabeth Gerber, Dr. Stephanie Fontana helped with the first paper, as well as Sarah Cook, and then Yuchun Chen was very instrumental in the gavaging. Casper Hansen did a lot of work for us with the chip-seq analysis. We got a lot of help for the metabolism study from the Vernon lab, and also I got a lot of help specifically from Caitlin Victor in the Vernon lab. I have to give a shout out to my old boss, Ronnie Cohn, who's up in Toronto now, but I'm grateful for the time I had with him. And I'd like to thank my thesis committee, Dr. Roger Reeves, Dr. Andy McCallion, Dr. Casper Hansen, and Dr. Hilary Verdon. I found that our meetings were very useful for me, and I gained a lot from them, and I really felt like they really helped me push my science forward, so thank you for that. And I also want to thank the Human Genetics Department. I want to thank Dr. Valley for always helping me, and especially in my time of need when I was looking for a new lab. Really instrumental in helping me, and I'm always be grateful for that. Dr. Kirby Smith as well, he was helpful there. He was giving me lots of advice right before my orals about finding a new lab, and I just felt like everybody in the department really had my back, and I really appreciated that. And then also with that, I really want to thank Sandy. She as well always had my back. Anytime I needed anything, she would always help me out. Apparently, she had a bowl of stress candy for people to take some. I never had any, but I think it's good. I think it's good that such a thing existed. And that's it. Thank you.
Questions? Joel, I think my brain is still back on about slide 3. You talk a lot faster than I can think. But can you show us which of the corrected models did you show us behavioral data?
What do you mean?
So the Morris maze, was that corrected with EDU or with the AR42?
Yeah, so the Morris water maze was corrected both with AR42 and as well with the ketogenic diet.
And when you stopped giving the drug, did they lose that?
That's a great question. So that's kind of one of the next experiments that we're getting into the lab, which is everything we've done, which has been 2-week treatment and then sacrifice the mice for analysis. So I couldn't see if they got better. But that's one of the next things. Two different ideas that I've been thinking about. One is treat for 2 weeks and then stop and then see if they keep it up. And then the other one is just I had another one, but I'm gonna stick with that one. Yeah, so that's a great point. So
which also brings up the possibility that what you're looking at is an immediate impact on synaptic function.
Yeah, so
based on the.
So the idea that we have here, so other groups have studied this methyltransferase, and what they seem to think is that it really affects, stem cell populations and helping them differentiate into adult cells by opening the gene expression there. So what we think is happening is that there's a stem cell population in the hippocampus that's not being expanded, and then when we give this drug, we're rapidly increasing that. And so there's two ideas: that is that going to fix it for life, or are we just kind of jump-starting the system, but then after a period of time, is it no longer going to respond and kind of peter out? So we haven't looked at that, and that's, I think, going to be an important thing to look at. Proceed.
When I first saw your increase in the NADH, NAD+, I was first thinking that maybe, I don't know which ACE stacks are involved with this KMT2B, but like a lot of them are all like NAD+ dependent enzymes.
Yeah, so that's actually a great question that I had been thinking about. I actually have thought about it. I'm not just saying that. It's actually, so that's one thing. So are you talking about like the sirtuins, which are, yeah, so sirtuins are NAD+ dependent. So that's another idea I thought about. I didn't want to include in this, but basically if we have a shift, so sirtuins are used by, are a class of HDAC inhibitors that are going to simply put decrease open chromatin. And if we have higher NADH, then there'll be less NAD+, which means the sirtuins will be less active. So in that way as well, it might promote more open chromatin, if that's what you're asking.
Yeah, that's right.
Okay, cool.
So if I were to go on
Seriously?
So if I were to go on the ketogenic diet now, would I produce enough beta-hydroxybutyrate to affect my epigenome?
Well, I mean, do you have Kabuki syndrome?
Well, like you showed that the wild type also increased beta-hydroxybutyrate.
Yeah, they do, but...
So could that affect my epigenome if I produced a whole bunch of beta-hydroxybutyrate?
I guess it potentially could. I mean, although what I saw here was that it didn't seem to have much of an effect on wild types for increase of H3K4 trimethylation or anything else, but our in vivo work in cell culture, when we really increased beta-hydroxybutyrate levels, where we did see an increase, so it's potentially that you might have to be on it longer than it could have an increase, but my data suggests that it might not have an effect on you.
And then one more thing, the AR42 versus the ketogenic diet, have you done just like expression, like a microarray looking at AR42 versus ketogenic diet?
So I didn't do one comparing those two, but I do have data from microarray. It was actually done by Janae in my lab, helping me out with my project. this is awful. It's frozen. I can't answer your question. I'm sorry. I guess my hands are tied, so my bad. Yeah. Here we go. All right, you're lucky. Just load. Okay, so we did a microarray. This is first just showing just the mutants, KMT2D mice compared to the wild types. And this is just a microarray of gene expression to see if we could find a set. And this is kind of, I didn't want to include this in my talk because I didn't want to get into specific genes, but we did do this work. And you can see here that there are a set of genes that are downregulated in the KMT2D mice. You can see 57 of them, which we defined as significantly downregulated, and only 2 that were increased. And there's a lot of genes here, and a lot of them do have roles involved in neurogenesis and also oxidation and processing, fat processing. But
I guess my question is, have you looked how that would change with both AR42?
Yeah, so what we did was, where is the data? So of these 57 genes, we found that I think 5 of them were able to increase in mutants on the ketogenic diet treatment. I have that data here. So this is just qPCR from that. We were able to see a rescue for these. We were able to see that they were decreased in the KMT2D mice, and then they were increased in the ketogenic diet treatment.
Have you considered knocking down or knocking out those genes to see if you can reach?
Yeah, so I mean that's one idea. So an alternative idea of treatment, instead of trying to look at the epigenetics, we could just target the gene pathways, figure out exactly which pathways are affected, and then give a drug that's targeted to just those pathways, and not even worry about the epigenome. Because one thing that I kind of didn't bring up is that massive change, trying to affect the entire epigenome, is going to have unintended downstream consequences, which is another barrier to go into the clinic. So yes, one idea is if we can look for these gene pathways and target just those, we may be able to have a similar response. Yeah.
Any thoughts on why just happenstance is enough in the KMT2D?
Sorry, can you say that again?
Oh, any thoughts on why just happenstance is enough to cause Kabuki syndrome with KMT2D?
Oh,
I would think maybe.
Why just one loss of copy is enough?
Like maybe there's other.
Yeah, so that's a good question. So there's a lot of other diseases that in the lab we call the Mendelian disorders of the histomachinery, which are other proteins that are involved in the histomachinery, some that add acetylation, some that remove methylation, that all are dominant, where they all have just one loss of function. And what's interesting actually is that you can't even have a homozygous. So a homozygous is actually lethal. I have some, this is just embryos. And we're trying to breed to get homozygous mice, and it appears that these are lethal at about embryonic day 8 or 9. And some other groups has looked at that as well.
So is that the only H3K4 methyl?
No, that's the thing. So there's multiple H3K4 methyltransferases, but for whatever reason, it's non-viable in a homozygous knockout, and just the one knockout itself is in effect. So what we think is that these things are very tightly regulated, and even just losing half a copy is enough to really throw this whole system off, and clearly 2 is lethal, so that completely destroys the system. And even though there's redundancies, there's many different genes that add H3K4 trimethylation. That is kind of one of the mysteries that we thought about, is why just one loss of half a copy of one of the many methyltransferases causes such a phenotype.
Are the other ones like double knockout, like non-viable too?
No, yeah, pretty much almost all of these are dominant, where the homozygous is lethal, and then they're all seen in dominant cases like that.
And then my last question: do you see any decreased activity of the erasers, like as a compensation unit?
I didn't look specifically at erasers to see that. I also looked at acetylation, which is another marker of open chromatin, and I actually saw a decrease of that as well in these KMT2D mice, which further confirms their idea that open chromatin systems are linked. But I didn't look specifically at other erasers. Yeah.
You had the CHIP data earlier for H3K4 trimethylation, and your mice were, it was the AR42 treated mice, you saw an increase in H3K4 trimethylation, that's right?
Yeah.
I'm curious what fraction of the promoter-associated H3K4 trimethylation can account for that increase? Did you take a look at that?
Yeah, it's a good question. I don't remember it exactly off the top of my head, but I think a lot of those were in promoter region. Much of those peaks were in promoter regions, because H3K4 trimethylation is primarily located at the promoter regions.
But the increase at those promoters, it would account for the majority of it. Wouldn't be any H3K for that set of promoters.
I think so, but I'd have to look more closely at that. All right.
There's actually a lot of the ones that change quite a lot are outside of promoters.
So I think this is on your list of planned experiments, but I just want to make sure I got it straight.
Yeah.
you haven't had a chance to look at different ages to see if the response is the same in mice of different ages. Is that right?
Kind of. So we actually did in our first paper. So the data that I showed you, every single mouse was always the same age, which was between 1 to 2 month old, which is considered young adult. We always wanted to. One of our experiments we wanted to do was to try to get younger. The problem is you really can't do some of the behavior tests at mice much younger than a month, just because they have to be able to actually swim. But we also did look at 5-month-old mice, just at the neurogenesis. And while all neurogenesis, as your age decreases, it did appear for AR42 that we got a rescue as well, although it wasn't as good at the 3-month point. So we're really thinking, and this kind of is confirmed by other studies with just studying neurogenesis, that there is going to be more of a critical window, and the earlier you hit it, the better it's going to be. One thought that we had was if maybe we could treat pregnant mothers with the ketogenic diet, that maybe we could in utero help the potential pups, but that's a little bit of ways away to design that.
And I suppose there's two issues. One is the neurogenic response, and the other is whether or not more neurons actually does anything to your behavior.
Yeah,
and
that's a good question.
Do you have any thoughts about that?
I mean, there seems to be in the field data that goes back and forth. It seems like it kind of seems like there's a threshold point where if you're below it, it's hurtful, but keep going more and more isn't necessarily going to be more helpful. So I'm not exactly sure. They might be just below this threshold, but I'm not exactly sure at what point do you have to get it up to that it's going to be all right.
And have you looked at any other populations of neural precursor cells that might also have the same sort of downregulation in Kabuki syndrome and then respond to it?
Yeah, so I personally haven't. One of the grad students in my lab, Giovanni, that's actually his project, is to look at some neuronal precursor cells. Another population that has high expression of KMT2D and has kind of been associated with this is in the cerebellum, but I mostly stayed away from that in my studies. But that could be another potential area to look at.
And are there any clinical observations that suggest that in the patients themselves that this kind of response might be possible?
You mean like anecdotal evidence or just... So I haven't interacted much with the patients, but from what I've heard so one thing that we want to do, we haven't actually put any patients on diet or on the treatment. One thing that we want to do is look at to see if any patients are already on the ketogenic diet to see what effect that might have. I think Hans told me an example of a mother who said when she decreased the sugar for her child, he seemed to do better, but that's pretty anecdotal. I don't know, did you have another example of this?
Well, there are people that, I think there are probably people that will try the ketogenic diet once this paper comes out. We're not suggesting that at this point. But it is interesting to look at the development of these individuals because many of these adults have done much better than people expected early on in life. So there are adults where, when they were children, the mother was told this child is never going to talk, they're never going to do anything, and the child ended up going to college and doing much better than anyone expected. And there's actually a lot of things that can stimulate neurogenesis. Exercise can stimulate it, learning can stimulate it. I think that would be some anecdotal evidence that maybe this phenotype is not as fixed as maybe some other causes of potential disability.
So have you looked in patients and just looked through their beta hydroxybutyrate levels to see whether that itself is a modifier of the phenotype?
Yeah, we have not yet. But this is relatively new. The last maybe 3 to 6 months has been kind of uncovering this metabolic phenotype.
Yeah, it's fantastic work. It seems like the application of it is very clear. A couple steps forward, pretty clear with each.
Yeah,
it's great.
But we're hoping to do that kind of going forward.
Good. All right. So thank you guys for listening, and I just have a little bit more. I'm going to go to my acknowledgments.
This is cleaned up.
Yeah,
there's nothing in here about the 49ers, is
there? There might actually be. You might just have to wait and see. So I just want to, when I was first getting these slides together, I had this kind of silly idea where I was going to say, I want to thank the person who's most important for my work and just show a bunch of pictures of myself. I didn't know how well that was going to go over. And the more I thought about it, actually, the more I realized that how much help I've had from so many people and how so many people at Hopkins, both here and throughout my life, have really had a positive impact on that, and I just want to take this, next 10 or so minutes just to thank everybody. And I'm gonna try to keep it short. I'm gonna try to not thank every single person in my life, but if I keep going long, you can just tell me to stop. So I have to start off. All right, I guess I'm done. So I gotta first start at the top with the boss, Hans. So this is how you know him as respected MD, PhD at Hopkins, leader of the epigenetics. Leader of the epigenetics and chromatin clinic at Hopkins, but there's also another side to him you don't know about. He's still recently removed from grad school, and still likes to have fun. I'm not exactly sure what they're doing here.
It's the T-bone. How do you not recognize that? I
didn't want to assume. This is just I'd rather not try to guess what's happening here, just to stay away from that. But in all seriousness, I do actually want to say a big thank you to Hans. Hans kind of brought this up at the beginning, but I'll just tell the story a little bit more closely. In the middle of my second year, my boss, Ronnie Cohn, he accepted an offer to go to the University of Toronto, and after talking with him, I decided that I was going to stay here, and at that point, I was homeless, and I had to find a new place. And Hans didn't even have a lab yet, but I was talking to some people, and they said that, he was starting up, he was interested in doing some of the translational work that I was interested in, and he didn't have space yet. He didn't even have a signed offer yet. But I went to him and I said, hey, I want to work with you, I want to be in your lab. And instead of saying, oh, I don't know if I can take you or this, he put a lot of faith in me, and he said, this sounds great. We talked about ideas, we just had, great interaction, and it was just the match made in heaven. Seriously though, he showed a lot of faith in me by taking me on so early. He had, hadn't even been set up yet. We got to the lab, we didn't have anything, and we were able to build the lab together, and then with everyone else. So I was just really appreciative that he gave me that opportunity, and I hope I did right by him and made him proud. So now getting to the rest of the lab. Here's Li Zhang, I mentioned her. She's like half lab manager, half lab tech, half postdoc, half something else, many different halves. But she's just amazing. She basically does the work of 3 or 4 people and really keeps our lab running. When she was gone this summer, everything is kind of, yeah. But she's just amazing. Anytime I need help with anything, she's the person I go to, as I said. If I need help ordering, she's the person. If I need help with an experiment, she's the person. And she's just always been really supportive and helpful, and I really want to say thank you. And then also my 2 lab mates. These are the 2 new grad students who joined just at the end. I kind of feel like a big brother that got some little siblings right towards the end, so even though he's actually older than me. This is Gio. I'm not sure why he's wearing the helmet. I've seen him wear it before though, so maybe it's a thing. I don't know what's going on. And here's Janae. So, like I said, these are pictures that I just took from their Facebook. I didn't even have to bribe anybody, so I think the lesson to be learned here is don't trust social media. But no, they've been really great. They were both played integral roles in my project, and it's been such a great opportunity not to just be the only grad student in the lab, to have other grad students for me to both mentor and still learn from. Has been great for me, and this last year that they've been here has probably been my favorite year just because of my interaction with them. I also want to thank Jo Farnar. She's been part of the lab for the past year and a half, doing some of her own projects, but also collaborating with Giovanni. And she's also, just like me, just giving me a lot of advice. She's always been so helpful and supportive, and I just want to say thank you to her. And, I can't not talk about my lab without showing the most awkward picture. So I just want to point out why this is awkward. So this is the picture. You might recognize it from my title slide. It was actually made by a Kabuki family, and this is supposed to be a Kabuki patient. It's got the symbol for Kabuki, it's got the Kabuki slogan and the Kabuki colors, and there's a butterfly, and the tear is supposed to represent the hardship of Kabuki syndrome, and he's looking towards the butterfly for the hope of the future. That's how Hans described it to me. Did I get that right? Yeah. Awesome. So we wanted to take a picture to thank them. They had given us some money, so he said, oh, let's take a picture in front of the MySeq. This is our sequencer. It's not even in our lab. It's in the lab next door. She said, let's put lab coats on. I just came from the gym. I'm wearing a sweatshirt. Gio looks right. He looks right in a lab coat. We just don't wear lab coats much. He said to leave a gap here so we could see the MySeq. Although maybe it was just because I came from the gym and she didn't want to stand next to me. It's like my favorite picture ever. Then I also, just talking about other groups, I really want to thank just my whole class in general. So, we came in together at 2010, and right from the beginning, the first thing we did is we always just hung out together all the time. We had lunch together, went to classes together, we hung out on the weekends together, and upper year students were saying, oh, that's cute, but this is not going to last. Good luck when classes start, good luck when you go to Bar Harbor, and they were wrong. And we still, we hung out together, we were very close-knit, and I think that really helped me just coming from the West Coast. I didn't know anybody here, and I felt immediately at home just with all you guys, and I just want to say thank you. You made my time here just great and awesome. And yeah, there's Haig there. This was our hike. I think it's Acadia Mountain in Bar Harbor. We had a great time there, went to all the classes, it was great. Sorry. I thought this was like a distraction. I read it. So speaking of groups that I want to be part of, as Hans mentioned, I kind of have a competitive edge. There may be a rule named after me at the athletic center about not being allowed to be violent during wrestling sports. I don't know about that. I don't know what I'm talking about. But I just want to show just some of the pictures of, some of the different sports teams I played. One here's softball, here's dodgeball, here's soccer. We just won the championship here. That's why they're holding me. And I just want to say, thank you to all these guys that have, been part of my life outside of school here. It's just, anytime I've had a bad experiment, you make me feel better. When I have a good experiment, you make me feel worse. So you really help me out, and I really appreciate that. And so, speaking of sports teams, I just have to get this off my back. So, estimate that I started watching sports when I was like 7 in 1995. So I lived in California from then until 2010. I had 0 championships. I was a diehard fan, and I got nothing. And then these past 5 years, I don't know why it says that again. It should be 2010 to 2015. That's wrong. I just had 4 championships, which is just ridiculous and unfair. And I'm going to move back to California now.
Wait, you're missing one. You're missing one.
I know. Exactly. And if you think about it, maybe it would have been 5 if I didn't cancel that Super Bowl because of the blackout. I may have had 5 in 5 years, so this is not fair. But never mind. I digress. So I also wanted to just show, also just not just my sports team, but also my friend group were, so helpful for me and just made my time here just so amazing. I think we all felt at the same time. Skiing, I just, the lightheartedness that we displayed is just amazing. I feel like I have jokes I can make, but I won't, because that would make him sad. And also, I want to thank my girlfriend Anna. She's always been helpful and supportive. Just even just going over these talks, she practiced with me as much as I wanted.
Like 6
such a great time. She just really also helped me through my grad school, and she importantly she would put up with me when I come home late. I'd like to say I was working late in lab, but I was probably just watching like a West Coast sports game. But always just putting up with that, and always just being happy and supportive, and that really meant a lot to me. So thank you for that. And then also, I just want to, thank my family as well. I wish I had some, cool story where I was the first member of my family to graduate high school, college, and grad school, but in reality, I'm just the 3rd Dr. Benjamin in my immediate family. So I'm not gonna get any awards for that, but I just want to say that you guys have always been there and supported me, and I'm, just really happy that you could be here to share this with me, and for everyone who's not here, shares with me. Thank you for your support over the years. And, for everyone who's not here, he's in these pictures or not in the pictures, or anybody who's ever impacted me, I just want to say thank you. You've played such an important role in my life, and I'm always going to be grateful. And I just want to leave you with the original title for my talk that Gio came up with, and that's it.
Bill, you did so good. Congratulations.
Congratulations, Dr. Benjamin. Thank you. Very nice job.
I'm going to stop.